Students, here we are going to discuss today another type of conventional vaccine, that is Salk Polio vaccine. Salk Polio vaccine used for to immunize children against polio. The poliomyelitis is a disease which is caused by poliovirus, which belongs to picorna virus group, where pico means small and RNA virus. So it's a small RNA virus, poliovirus. Okay. Now, the characteristics of the virus are it has a very small size, so the name pico, and because it's an RNA virus, we call it picorna virus. The size is very small, that is 20 to 30 nanometers. Having icosahedral or spherical symmetry, they are non-enveloped viruses, and hence they are resistant to various organic solvents. If you look at the etiology of the disease, poliomyelitis has been recognized as a paralytic disease of children. It is also called as infantile paralysis. The virus enters into the body by ingestion or inhalation of droplet nuclei containing the virus. The virus multiplies first in the lymphatic tissue or the alimentary canal. Once the infection is established, the virus then travels to the tonsil region and then to the Peyer's patches, and then enters into the blood stream via regional lymphatic system. The condition is known as viremia. When the virus enters into the blood stream, we call the condition, that is presence of virus in blood, is called as viremia. Now, through blood stream, the organism reaches the spinal cord. Once the organism reaches the blood, naturally fever develops. Through the blood stream, the virus reaches the spinal cord and then to the dura mater of brain. In the central nervous system, the virus multiplies in the neurons and destroys them by forming lesions in the anterior horn of spinal cord. Here, the virus destroys the motor cells, and destruction of motor cell leads to paralysis. What are the clinical features of poliomyelitis? The incubation period of the virus in the host body is 4 days to 4 weeks, but it may vary. Early symptoms comprise of viremia. After viremia, there is fever, headache, sore throat, and malaise. This may last for 1 to 5 days. This type of infection is also known as abortive poliomyelitis because it may not end up with polio. If the infection progresses, then fever comes again, which we call as biphasic fever, along with headache, stiff neck, and other symptoms of meningitis. The control over the nerves is lost, and condition becomes more severe, and it leads to paralysis of various organs, which causes polio. Now, how polio vaccine, Salk polio vaccine, is produced? Let us discuss. What is the organism used? Now, there are 3 types of polioviruses: type 1 poliovirus, type 2, type 3. All of these strains cause polio. Now, type 1 is Mohaney, type 2 is Lansing strain, and type 3 is Lean and Saukat strain. The 3 distinct steps, all the 3 virus strains are used for vaccine production. Okay, so it's a trivalent vaccine. The distinct steps which are involved in vaccine production are: first, cultivating the virus; number 2, inactivating the virus; and then testing the safety of the vaccine and desired potency. Let us discuss the method in brief first. In 1953, Salk developed a killed vaccine for the first time against polio. Salk polio vaccine is a formalin preparation of 3 types of poliovirus grown in monkey kidney cell culture. Okay, so it's a formalin preparation of 3 types of virus. All the 3 strains are grown on monkey kidney cell culture. All the 3 are highly virulent, and then all the 3 cell cultures are mixed, treated with formalin to inactivate the virus. And this is how trivalent polio vaccine is developed. The individual strain is grown separately in MKC. After providing sufficient incubation period for a period of 4 to 10 days, viral pools are extracted out. Virus pools of adequate titer containing definite number of viruses are then filtered to remove the cell debris, clumps, and other extraneous matter. And then the viruses are inactivated by treating them with formalin at 37 degrees Celsius for 12 to 15 days. Various stringent tests are carried out. to ensure that with strict specification, the vaccine production takes place. So very stringent tests are carried out to make sure that the virus has been completely inactivated, and freedom from extraneous matter has been obtained. The three types of viral pools are then blended to prepare a trivalent killed polio vaccine. Then it is tested for safety and potency. And after we acquire satisfactory results, vaccines are issued for use for the public. Now let us discuss the vaccine production in detail. What is the first step? Cultivation of virus. Now, viruses, as you all know, cannot grow on artificial media. So we require either tissue culture or living animals, animal culture. So here tissue cultures are used. So viruses grow readily on a variety of human and animal tissue cultures. Cultures of primate origin are generally used, like primary monkey kidney cell culture are universally used for the production of vaccine. For this purpose, kidneys are aseptically removed from monkeys and then ground into fragments. The tissues are separated out using various proteolytic enzymes like trypsin by a process called as trypsinization, which we are going to discuss hereafter, collagenase enzyme, etc. The dissociated cells of tissues are then thoroughly washed with Hank's solution of phosphate buffer, which contains antibiotics. Blood, fat, and other impurities are removed. The tissues are then chopped, minced using scissors, and once again washed. Then the individual cells that are separated by trypsinization are placed in a sterile glass or plastic vessel or bottle of various size and shape, which contain nutrient medium. Okay. And viruses are cultivated. Sterility is strictly observed at all stages of cultivation. So we cultivate the virus in monkey kidney cell culture. The nutrient medium used for producing monkey kidney cell culture actually contains whole range of amino acids, vitamins, growth factors. The nutrient mediums are also commercially available for cultivating viruses, like medium number 199 or Eagle medium. We prefer these mediums. To avoid contamination, antibiotics are also added to the growth medium. The tissue culture bottles are incubated at 37 degrees Celsius for 7 days. During incubation period, cells readily metabolize and grow. Actually, the cells, monkey kidney cell culture, the individual cells which are added to the growth medium, they grow and a monolayer of cells is formed. Then we inoculate the virus onto the monolayer. The viruses rapidly invade the kidney cells. After 4 days of incubation period, tissue cells get destroyed because of attack by virus. And slowly the tissue cells settle down, forming sediment at the bottom of the flask or bottle. The supernatant solution contains the viral pool are separated off, and then the sample are analyzed or assayed for viral content. So you prepare monkey kidney cell culture, add into a growth medium, inoculate with the virus, allow the virus to grow. The virus destroys the cells and the cells sediment at the bottom, and the liquid supernatant that remains on the top of the sedimented cell contain viruses. Okay, then the virus pool is extracted. The next step is inactivation and blending of the three viral strains. Inactivation of virus is carried out under great precaution and sterility is strictly maintained. Okay, we have to take every precaution that there should not be any contamination because of bacteria. One. Number two, all the virus, each and every virus particle must get inactivated. So the supernatant of tissue culture is passed through ultra fine membrane filter to remove the cell debris of tissue culture, which might interfere with the efficacy of vaccine production. Then formalin is added to the filtered solution to give final concentration of 0.01% in the virus suspension, which is thoroughly mixed and maintained at 37 degrees Celsius. Inactivation is completed generally within 4 days, but the process is continued for 12 days. In order to minimize the interference of formalin, second filtration is done on 6th day to remove the formalin. The inactivated single strain is stored at 4 degrees until the safety tests are completed. Same procedure is repeated for cultivation and inactivation of the remaining 2 strains of poliovirus. That means all the 3 type of strains, type 1, type 2, type 3 are grown separately. Virus pool is collected out separately and they are to be inactivated and then they are mixed. The final trivalent vaccine is then prepared by properly blending the 3 monovalent extracts or viral pool in appropriate quantity, and the contents are thoroughly mixed in rotating steel containers. We at every level, most rigid aseptic precautions are taken to avoid contamination. Excess formalin present in the blended mixture is neutralized by adding sodium metabisulphite, because each of the virus pool after filtration through ultrafine filter is treated with formalin and incubated for for a period of 12 days to inactivate the virus. Then on the 6th day, we again filter to remove the effect of formalin. Then finally, if any formalin is present after blending all the 3 viruses, we have to neutralize the formalin present by adding sodium metabisulphite. And then the contents are filled in ampules. The ampules are lyophilized, labeled, and sent for sterility and safety testing. Then they are packed and discharged into market. Okay. If you look at the flow sheet for the production of Salk polio vaccine, you can see here monkey kidney cell culture. Okay. This is monkey kidney cell culture, right? So cultivate the virus. Okay, you first form the MKC, cultivate the virus, right, in the growth medium containing MKC culture. All the 3 types of virus are grown separately in MKC culture, right? Then you extract out the virus pool, okay, and store it in cold room. Okay, how do you extract out the viral pool? By passing through membrane filter. Then you neutralize the virus by adding formalin and incubate at 37 degrees Celsius for a period of, actually within 4 days, the inactivation is complete, but incubate it for 12 days. And then on the 6th day, second filtration, and then store in cold room at 4 degrees. Then in this way, we have grown all the 3 viruses. So the virus pool is then blended. Okay, all the 3 viruses given proper same treatment are blended. After blending, safety testing, stirring and all, and then neutralization of the formalin effect is done. Okay. After that, the trivalent vaccine is filled in ampule, labeled, packaged, and discharged into market. Now see, what are the safety testings that are carried out? The following tests are carried out for checking the sterility, efficiency, and safety of the vaccine. First, we go for tissue culture test. So the vaccine or the samples are distributed in large number of bottles containing monolayer of monkey kidney cell culture, which is covered with nutrient medium. So we have monkey kidney cell culture in bottle covered with nutrient medium, inoculate it with the vaccine. Okay. After sufficient incubation, you examine the kidney cells under microscope for cytopathic effect. If poliovirus is living, if it has not been inactivated, either complete destruction of monkey kidney cell culture will take place or plaque formation will take place. Okay, and if plaque formation or destruction of MKC takes place, it indicates cytopathic effect, which indicates that virus is living. So it cannot be sent to market, right? So we have to send it back to the laboratory for inactivation of viruses. And in the tissue culture, if there is no growth, MKC is as it is even after incubation for a sufficient period, then the vaccine is safe to use. Animal inoculation. Monkeys are first treated with cortisone to increase their natural susceptibility to poliovirus, and penicillin injection is given to tackle the bacterial infection. Then the monkeys are injected with the trivalent polio vaccine, and they are observed for signs and symptoms of paralysis for a period of, say, you can say 2 days or maybe a week also. Within this time, paralysis may develop, and in severe cases, death may occur. If the vaccine is not efficient in controlling paralysis, above symptoms may appear. So the CSF from the animal is collected and searched for the presence of live virus. So if any paralysis takes place in the animal after animal inoculation, it indicates that the inactivation of virus is inefficient and we have to send it back for inactivation. Okay. So this is how safety testing is done. Now, for preparing monkey kidney cell culture, we have to perform trypsinization. So let us discuss trypsinization process. Poliovirus is cultivated in cell culture while preparing Salk polio vaccine, and usually we use monkey kidney cell line. Okay, the kidney of the animal is removed aseptically by giving anesthesia to the animal. The kidneys are washed in sterile hang solution. It's a well-balanced isotonic salt solution. After after washing the kidneys, they are fragmented into small pieces. The fragments are then immersed in 0.25% of trypsin, which is a proteolytic enzyme. We make the medium alkaline by adding 1 normal NaOH and maintain the pH at 7.4 to 7.8. Trypsin is a proteolytic enzyme and hence it depolymerizes the tissue fragments so that individual cells are separated. So when you mix the or keep the kidney fragments with trypsin at alkaline pH, the cells get depolymerized and individual cells are separated. The separated cells are stored in buffered saline at 4 degrees Celsius. They can also be stored at liquid nitrogen temperature in presence of glycerol, if not immediately required. Now, and as and when required, the individual cells can be used to form monkey kidney cell culture, mono layer of monkey kidney cell culture in the culture bottle. Now, we use a simple apparatus for this depolymerization of kidneys or for single cells, separating the single cells of kidney. Okay, this instrument has 2 side arms inserted just above the base. You can see this is an instrument. This is an inlet. Okay, and this is a rubber tube connected here. Inside, we place the fragments of the kidneys, and in the kidney fragments are placed in buffered saline with trypsin. After trypsinization, the kidney cells are depolymerized and individual cells which are lighter in weight get collected at the top of the buffered saline, and from the outlet, individual cells can be separated. The 2 arms of the small apparatus are connected by silicone rubber tubing. The equipment has an outlet at the top. The chamber is filled with buffered saline and trypsin up to the mark, which is near the outlet. Okay, fragments of kidney are allowed to enter from one side into the chamber. After trypsinization, the cells get separated and they float on the top of the medium. Fragments which are not trypsinized into individual cells are recirculated through the same rubber tubing for trypsinization. The process is repeated till all fragments or tissues of kidney get separated into individual cells. All separated individual cells are stored in buffered saline at 4 degrees and can be used as and when required. Okay, so this is all about Salk polio vaccine.